Welcome to the webinar on biological control of coal crop pests on the Central California coast by Diego Nieto of the University of California at Santa Cruz. I'm your host, Alice Formiga from the eOrganic Community of Practice at eExtension. You can find all eOrganic articles, videos, and our many upcoming and recorded webinars on organic farming and research topics on our website at the link on your screen. This presentation is being recorded, and you'll be able to find the recording on our website and on the eOrganic YouTube channel within the next 7 days, in case you'd like to view parts of it again or share it with others. We've also uploaded a PDF handout of this presentation, and there should be a handout section on your GoToWebinar control panel, and that's where you'll be able to find it. So before we begin, I'd like to just give you a really quick rundown of today's program. The presentation will last about 45 minutes, and following that, we'll have 30 minutes for your questions. If you have a question at any time, you can type it into the question box and hit return. If you don't see the question box open, just click the small plus sign or triangle next to the word question on your GoToWebinar control panel, and that will open it up. We'll be reading the questions out loud after the presentation is over, and we'll answer as many as we can in the time that we have. So today, I'm very pleased to welcome back Diego Nieto of UCSC. He has been a research entomologist at the University of California, Santa Cruz since 2001, and he's studied issues related to pest management in cotton, coal crops, strawberries, and olives. So thank you for joining us today, Diego, and I'm going to hand over the screen control to you right now.
Okay, thank you very much. All right. So let's begin. The project is about coal crops most of the time. The coal crop in question was broccoli. I'll try and specify when it wasn't. And there's a lot of economic information about broccoli in the state. California does a pretty good job of broccoli production. 95% of the country's total is produced in California. Specific to organics, that's about 91%. A lot of our research was done in Monterey County, which is just south of Santa Cruz. That county alone accounts for about half of all of California's production. So we're really in the heart of coal crop country, if you will. So the pests that we're going to be discussing, cabbage aphid, diamondback moth, and cabbage root fly. Obviously, they're specific to the coast, but they're also cosmopolitan, which is to say they're pretty much anywhere. You can find them anywhere in the world where coal crops are produced. And so there's a lot of similarities, a lot of applicability with respect to the different cropping dynamics that we'll be discussing. So some of the challenges with these pests, these are relatively specialized pests in that they only seek out brassicas. They only seek out coal crops. And so a lot of the dynamics that you could use with generalist pests to try and take advantage of preferences, those aren't always applicable here. Some of the crops, coal crops like Brussels sprouts, are in the ground for a long period of time, which means they're very ecologically apparent to these specialist pests. Some of the plant structures offer refuge from insecticides. It could be aphids on the underside of a relatively rigid leaf like a broccoli leaf, or seeking shelter inside of a floret or a Brussels sprout. And contamination threats, be it from an aphid or a caterpillar, lead to pretty low action thresholds with respect to these applications. So I mentioned Monterey County, where we produce a lot of coal crops, but just to kind of reinforce the notion that these are global crops with global pests. So there are coal crops that are being produced all over the world, and the U.S. is present, but not necessarily a leader. Okay, so let's begin with cabbage aphids. Just about anywhere you have a coal crop being produced, you've got cabbage aphid, which is more often than not the most problematic insect associated with this crop. Capable of very rapid reproductive rates. So a winged aphid will fly into a field. This is a female, and she is capable of asexual reproduction, producing anywhere between 30 and 50 offspring. These are all cloned females. They'll grow up and produce 30 to 50 offspring, and so on and so forth. So the reproductive rates, the growth rates of the aphid population are very, very quick. With respect to cabbage aphid biological control, there's a pretty big community of insects and spiders that are capable of occasionally making a dent in reducing cabbage aphid colony sizes. However, most consistently, most commonly, it's syrphid flies. These are true flies in the Syrphidae family that are considered to be the most economically important or relevant predator community. Each larva can consume hundreds of aphids. So in case you're not familiar with the life cycle, a syrphid fly will lay an egg that'll hatch out into a predaceous larvae or maggot that'll consume lots and lots of aphids and then pupate into another fly. There's a very diverse community of syrphid species on the California Central Coast. Let's take a look at. We're going to take a look at a quick video here, just to give you a sense as to a syrphid larvae in action. So this is a very immature syrphid larvae, and it is searching out for lunch. So you've got some cabbage aphids there, and you can also see above on the top left-hand corner, there's another very, very, very small syrphid larvae as well. Anyway, so the syrphid larvae goes. It's going to make contact with an aphid, and it's going to start feeding. There you go. Thank you, Alice. Okay, so table 1 here shows the syrphid species that have been found in California, at least to the best of my knowledge. There were different studies. The first 3 columns, or the 3 columns on the left, were conducted on the Central Coast, including Monterey, Santa Cruz, and San Benito counties that are in broccoli and lettuce. The 4th and final column was in cabbage in Orange County back in the late 60s and 1970s. There's a total of 13 syrphid species that have been documented. Most commonly, at least with respect to cole crops, you have Allegrapta obliqua, which was ranked as the most common syrphid species in the 3 cole crop studies. And beyond that, the similarities start to fall apart a little bit. But you do have a very broad community of syrphid species that are found in the state. So we'll describe a few of these syrphid species that we encountered on the coast in cole crops. So we'll start with Allegrapta obliqua. Again, this is the most common species that we found in cole crops. It's found all throughout North America in a very wide array of cropping systems. So it's by no means a specialist. In fact, I should point out that all of the syrphid species that we're going to look at slightly in depth are found in places other than California. These larvae can consume lettuce aphids at rates of 22 aphids a day, or 228 per lifetime on average. That's based on laboratory studies done by Hopper et al. And the appearance varies considerably from obliqua larvae to obliqua larvae. They can vary in color, and Alice, we want to fire up the next video here. They can vary tremendously. They can be a bright green. They can almost be sort of an orange color here, which is what you see. This is a mature Allegrapta obliqua larvae. They have 2 bands, 2 dorsal bands that run from the spiracles in the back to the front. Relatively smooth texture, not a lot of bristles or hairs. You can see it's starting to chow down on this particular cabbage aphid here. Thank you. Next, we have surface opinator. This has been observed ranging from southwestern Canada down to California. It's been a prominently described woolly apple aphid predator in Washington. In the same study, it was apparently it benefited from sweet alyssum beneficial insectaries that were used. This has a very different texture than the smooth texture that you just saw with obliqua. It has a lot of spines and ridges. It has V or chevron shapes on the back, on the dorsal side, that are pretty helpful in distinguishing this species. Let's take a look at the next video. This is a short one. So here's a surface opinator feeding on a cabbage aphid. And hopefully you can see the V or chevron shapes that are present. Next, Eupiodes americanus. This is widely documented throughout the continental U.S. It's apparently been very helpful in managing woolly apple and rosy apple aphids. This is a bit enigmatic in the sense that in a given year it can be one of the most abundant syrphid species. It's tremendously effective at knocking down aphid populations. And so you begin to feel really great when you see it, and then the next year it's just gone. It's not present at all. And when I say not present, I mean multiple farms and multiple counties, and it's just not there. So it's not, for whatever reason, it's not a species that you can count on year to year. And this was backed up in some of the apple publications that have been present, that it's tremendously effective when it's there, but it's just not always there. It has an opaque tortoiseshell type pattern and a dark dorsal heartline that's interrupted by transverse bars on the back half of the larva. And I should also point out, when it is present, it is by far and away my favorite. If it's fair to call a maggot charismatic, I certainly would. These things will be happy to hop up on your hand and sort of move around and say hello. They're really pretty cool to work with. Next, Sphaerophoria sulfripes. This, at least in documentation, has a more narrow range. That's been documented in California and Oregon. This has, I guess, what I would describe as an anticipatory oviposition strategy, which is to say that relative to the other species, they will lay eggs. The female syrphid flies of the species will lay eggs earlier and with fewer aphids present than the other syrphids in that community. And because there are fewer aphids that are available when those eggs produce small little larvae, the larvae end up demonstrating lower predation rates. In other words, they consume fewer aphids, and they grow to be smaller, mature larvae than the other species. And that's based on the work done by Hopper as well. It can be tricky to distinguish these larvae from Allographta obliqua. They look very, very similar. The most helpful distinguishing characteristic between the two is the breathing tubes. These are fused all the way to the top in sulfripes. In Allographta obliqua, there's a Y shape. There's a divergent sort of apical end of the breathing tubes that is helpful in distinguishing these two species. And last but not least, Platycheris stegnus. This is pretty unique. It's documented in the western portions of Canada, the US, and Mexico. And contrary to sulfripes, this has a very high density oviposition strategy, which is to say, if the previous syrphid species likes to colonize a field very early in the process, this does the exact opposite. It waits until aphid population densities are very, very, very high, and then it goes in with multiple sort of clusters of eggs and really cleans things up in a hurry. These larvae are pretty distinct in that they're an orange or rust color. And on this next slide, you can see these very small, very immature larvae. And they almost look like little orange worms. They're very, very small. But given the kind of gray, dull green color of the broccoli leaf and the gray color of the cabbage aphid, very small larvae can be quite distinct, very conspicuous because of that bright orange color. And so you'll see multiples of 2, 3, 4, 5 of these little larvae smack dab in the middle of hundreds and hundreds of aphids, and they're there to clean things up. So they don't arrive until very late in the growing season, but they can clean things up really quickly. In a matter of weeks, they can really change the course of an aphid population. So these images and information and more are available in a Syrphid larvae guide that's available on the CalCore network. The link is listed here if you're interested. So let's evaluate the biological control services that are provided by the Syrphid, and we can do so using some exclusion studies. Two different strategies for this. The first is physical, in other words, caging, and the next is chemical, using an organically compliant larvicide. The advantage in doing this is that it provides an ability to quantify the benefits of predation, which is difficult to do otherwise. And then also, because aphids are very immobile, it makes it pretty convenient to work with these critters as well. So I'll first start with the cage experiment. This was done at the Center of Agroecology and Sustainable Food Systems at UC Santa Cruz. We just used PVC pipe and insect netting to literally cage a broccoli plant, exclude other insects from coming in. We would then inoculate or place single alate aphids on both caged and uncaged broccoli plants that had been cleaned and removed. Other insects had been removed if they're present. And then we determined harvest at the end of the growing season by the presence or absence of aphids in the broccoli head or floret. So here's an image of some caged broccoli plants. And when you compare the harvest rates of caged and uncaged plants, you can see there's a 33% increase in harvest when you included the opportunity for predators to come in and manage aphids. In the Larvicide experiment, this was done more recently at the same farm at Cassvus. And here we had Entrust, which is an OMRI-improved larvicide, and that was applied to half of the broccoli plants. Only once in the growing season, after we had started to observe some Syrphid larvae that were beginning to colonize the field. So here's Cassvus again. Here's our test plot. We have 4 different rows or reps of broccoli. 2 were randomly selected, and trust was applied. Again, this is only once during the growing season. So on this graph, you can see the top is 2012, 13, and 14 as you go down. The x-axis is the dates. The y-axis is the mean number of Syrphid larvae per plant. And the arrow, which is on the left-hand side of each graph, is the point at which Entrust was applied. Again, it was only applied once. The blue line is the insecticide-treated plants, and the yellow line is the untreated control, or plants that did not receive a larvicide application. And so what you can see is that by applying Entrust, that bought us 1 to 2 weeks of kind of Syrphid larvae knockdown. In other words, there were 1 or 2 weeks, depending on the year, in which there were differences between Syrphid larvae abundance in treated and untreated plants. But by the end of the year, the Syrphids ended up catching up. In terms of how this translated with respect to aphid abundance on these same plants, so again, the arrows indicate when the Entrust was applied, and you can see that while aphid densities were comparable at the time of the larvicide application, by the time they were harvested, there were significant differences in the sense that plants that had the entrust applied to them were exposed, and the syrphid larvae were suppressed, even though only momentarily, ended up having higher aphid densities on the plants. And those differences range from an average of 72 to 190 plants. I'm sorry, 190 aphids per plant. So if we switch gears a little bit, so we know that there's a benefit to predation, to syrphid larvae predation. If we look at some of the temporal dynamics that begin to influence those benefits, in other words, the timing that's involved in a coal crop being produced, and a couple of different things that you can take into consideration. So first, syrphid larvae, they consume a lot of aphids, right? But if they consume a lot of aphids, that means there have to be a lot of aphids present in order for them to consume, and that takes time. And you can then add in the fact that many of these coal crops, not all, but many, have a relatively short turnaround time. Broccoli, for example, is more or less 10 weeks from transplant to harvest. So there are some dynamics that need to take place in a relatively short period of time. So here's a theoretical organic broccoli field. We have time from transplant to harvest on the horizontal or x-axis, and insect abundance on the y-axis. The blue line is aphids and the red line is syrphids. So aphids are going to colonize a field and rapidly increase their population size. Syrphids are going to respond. As syrphids respond, then they're going to begin to bring down that aphid population. And hopefully this all takes place in time for harvest. Now this is all presumably predicated on some kind of aphid egg-laying threshold that needs to be met in a timely fashion. And obviously this is going to be different species to species, but presumably there's some kind of threshold that needs to be met. Here's some work that was done by Ambrosino, Jepson, and Luna a few years back, looking at this issue of egg-laying behavior by syrphid species when comparing different numbers of aphids in a given plant. So the top graph is the number of syrphid eggs per plant, and the x-axis has a number of aphids per plant in different categories. And so you can see that the number of hoverfly eggs or syrphid eggs don't really get consequential until at least you get above 50 aphids per plant, right, at the very least. And over 100, it's even better than that. The bottom is the percentage or the proportion of plants with hoverfly eggs. And again, you need at least 50 aphids to start producing some kind of effect from syrphid flies, from females. So that means that the timing is really important, right? So here's an alternative theoretical broccoli field. If the aphids are late arriving in your field, or if there's an insecticide application made early in the growing cycle to suppress that development chart, that means that the aphids are going to hit that threshold later in the season. It could be 50 aphids per plant, 100 aphids, 200 aphids, whatever it is. The eggs are going to be laid later. The syrphid larvae are still going to do their thing, but it might be too late to knock down aphids in time for harvest. And so at that point, you get lots of aphids in the floret or in the curled up leaves, and so you have a diminished harvest. So the timing is very, very important for when all this takes place. So we can look at some experiments that begin to investigate these issues, these dynamics. We can look at sort of a controlled situation first when we exclude predators, just to make sure that our baseline understanding is confirmed. So in other words, there were different inoculation times during a broccoli growing season when and where predators were excluded. And the earlier you introduced an alate aphid to a plant, the worse off that plant was with respect to harvest. So you can see when aphids were introduced to broccoli plants early in the season, they had very, very low harvest rates, and those harvest rates went up the later the aphids were introduced to those plants. This is when you exclude predation. If you compare that caging experiment to field experiments where predators are obviously out and about, and we might get a different look. So these are 4 different organically certified farms in Monterey County. And this is a summation of those farms. The x-axis is the aphid arrival time, the number of weeks post-transplant. So in other words, on a given plant, when did an aphid first arrive on that plant? And based on that arrival time, how many aphids were there at harvest on that same plant? And that's what's on the y-axis. And so what you can see is the earlier an aphid colonizes a plant, the fewer aphids are ultimately on that plant at harvest, at least based on the first 3 weeks of the season. The period in which there are the most aphids is the middle portion of the growing season, weeks 4, 5, and 6. And this might seem counterintuitive, but we can overlay the syrphid larvae abundance during the course of the season that's available on those same plants, and again, based on when aphids first colonized a plant. And if you think about it this way, it might make more sense. So the earlier aphids arrive on a plant, you're giving syrphids more time to locate those colonies, lay eggs, hatch out, and eat those aphids in time for harvest. When aphids arrive very early, then there's plenty of time for them to be eaten up. When they arrive very late, there's not a lot of time for them to build up their colony size and infest a floret. So it's really in the middle that it becomes problematic, right? They have enough time to build up their colony size and infest a floret or a leaf, but not enough time for syrphids to come and gobble them up. If we want to remove the aphid density histogram, keep the syrphid larvae line, and add the harvest line, the harvest rate line, for that same year, they overlap really nicely. So again, the more larvae you had, the higher harvest rates you had. So harvest is in the red line and is represented as a percentage on the left-hand side y-axis. At the bottom, you can see if you just on average look at plants that were harvestable and compare them with plants that weren't harvestable, again, based on aphids that were present or absent in the broccoli floret, aphids arrived earlier, about an average of 2 weeks earlier, in plants that were harvestable, and 2 weeks later on average when plants were not. This is for 2002, 2003. So I'm not, it would be nice if all of the years were always as clean as the data I'm showing you. They're obviously not. But this is one sort of scenario that might help explain some of the dynamics for aphids and syrphids in organic cold crops. So just to summarize, with respect to when aphids arrive early and mid and how that contributes to harvest, in the cage experiment, right, the earlier they arrived, the worse things were. But in the field, when predators were available and effective, the earlier aphids arrived, the better off you were. So moving forward, I think it's really important to, and I actually haven't touched upon this yet, address the inconsistencies that are present with the predation services that are provided by syrphids, which is to say, when they show up and when they show up on time, they're wonderful. They're very effective, but they don't always show up or show up on time. So that means we need to address some of these kind of fundamental questions about why aren't they always showing up. So for example, what affects the pull dynamic or the attractive dynamic into cold crops? Microclimates, coast versus inland or season, summer versus winter. And what generates that pull into the first place? Nutritional opportunities versus reproductive opportunities, or food versus sex is another way to think about it. Is it the addition of sweet alyssum or some other beneficial insectary that's providing improved feeding opportunities that's attracting them into the field? Or is it reproductive opportunities? In other words, lots of aphids and lots of great opportunities to lay eggs, or some combination of the two. There's been some good work of late looking at how to optimize beneficial insectary spatial arrangements, both in terms of sort of how it's arranged and how much you need. This has been done by Eric Brennan. And he seems to suggest that you can still maintain a good Syrphid biological control with less sweet alyssum. And that might indicate, just maybe, that it's the reproductive opportunities and not the nutritional opportunities that provide a pull into a field. And that's just a guess, if that were the case, then it might be wise to examine some alternate aphid prey candidates to try and figure out how you can provide some pull in ways that don't affect your yield, to get those syrphids into the fields earlier. Okay, switching gears. Let's move on to the Diamondback Moth, Plutella xylostella. The Diamondback Moth is on the left. Cabbage looper is in the middle. Imported cabbage worm is on the right. It's really common to see these three caterpillars in the field. A Diamondback has some distinct black bristles, forked posterior prolegs, and is very squirmish, by far and away the most squirmish of the three. Some of the challenges associated with this pest, they are infamous for their ability to develop insecticide resistance. If memory serves, they were the first to develop resistance to DDT. They've also developed resistance to BT and spinosad. And also the risk of contamination, particularly in crops like Brussels sprouts, makes it challenging and creates low action thresholds. We were interested in looking at biological control of Diamondback on the coast. And Diadigma insulare, which is nyctomonid, is the parasitoid that's most well known in North America. So there actually end up being very few records of Diamondback moth biological control in California. There was one that was done by Oman and Plattner in 1969, which talked about Diadigma insulare being present in Southern California cabbage, commercial cabbage. And the only other that we could find was done by Fox et al. In 1990 at CASFA, so UC Santa Cruz, but there weren't any sort of commercially relevant publications on the coast. So we set out to see what was actually happening in commercial ag. So we have multiple collection sites up and down the coast. And here's sort of an overview of what we did, what we found. So the N is the number of pupae that were collected, and the percent parasitism from each particular site. So there were more than just 2 sites, but those are some of the highlights. So for example, in Salinas in 2012, there were 16 pupae that were collected in the season, pretty low pest density. But all 16 that we collected were parasitized. Overall, in the region for 2012, 61% of the Diamondback moth that were collected were parasitized. 55% in 2013 and 70% in 2014. That's all broccoli. All of the previous table that I just showed was all broccoli. If you look at Brussels sprouts, and these are unmanaged organic Brussels sprouts, so there was never an insecticide, there was no BT, and nothing was applied. Then the population densities of Diamondback moth get much higher. So this was in 2010, and you can see that the Diamondback densities get pretty high. They peak at just about 3 Diamondback per plant. But parasitism is pretty high as well. So So it peaks at a little over 70%. So the parasitism that's being provided is pretty sufficient. And by the time you get to the end of the season, then the diamondback moth is knocked down. 2011, this is the same field, unmanaged organic sprouts. The diamondback moth densities per plant were much, much lower, right? So we had just about 3 diamondback per plant in 2010. Here we have 0.3, so a lot less. And percent parasitism is lower as well. You get 150%, but it's quite low. When you look at the yield and the possible contamination that that could have taken place in this unmanaged field, we have three different crops that are being looked at in 2 years, 2010 and 2011. A total of 1,162 sprouts that were opened up and looked at, and only one had a diamondback moth larvae or pupae. So the sort of physical contamination was almost completely absent from these sprouts. So a couple things to think about with respect to diamondback moth biological control. The parasitoid Diadegamous is very density dependent. So in years where you don't have a lot of host density, you're not going to have a lot of parasitism. This means it's possible that sprouts that are some of the first cycles, the earliest cycles of the year, they might not benefit from as much parasitism as sprouts that are a little bit later. Diadegma is also very susceptible to insecticides. So anytime you begin going in with even an organically compliant larvicide, you're going to knock down this parasitoid. And there haven't been, at least to my knowledge, any efforts to see what beneficial insectary habitat or other conservation beast measures on the West Coast could be taken to benefit this parasitoid. And we'll conclude with cabbage root fly, Delia radicum. We just started working on this pest a couple years ago. This is when it's sort of present, it's particularly challenging. It's active year-round. The insecticide or chemical treatments there basically aren't really any in organics, and in conventional they're limited. And we really don't know very much about it, particularly with respect to biological control on the West Coast. There's very, very little that's known about it. If you're not familiar with this pest, it's a true fly that lays eggs at the base of cole crops. Those small larvae hatch out and feed on the roots of the crop and pupate in the soil. So in addition to just sort of the inherent interest we have in biological control of the cabbage root fly, part of what I'm about to share resulted from a need to maintain a cabbage root fly colony, which you can see here. So we needed to maintain this colony, which ends up being a lot more challenging than you might expect. And we were field collecting a lot of pupae, pupae from organic farms in order to supplement the colony. And there'd be periods of the year when the vast majority of pupae that we collected ended up being parasitized by braconids. There was one field we collected 400 pupae, and the vast majority of them were parasitized. So it was frustrating from the standpoint of trying to maintain a colony, very exciting from the standpoint of biological control. So we did a survey in Santa Cruz County on different organic farms, looking at the biocontrol that may be present from field collected cabbage maggot pupae. And they're collected during different times of the broccoli season, early, mid, and late. So in this particular graph, this is from the summer of 2014. This is rove beetle parasitism. And there's none in the early portion of the season, as you might expect. A little bit in mid, and it starts to tick up a little bit in at harvest. These next graphs are from this year. They're from the spring of 2015. This is in cauliflower, organic cauliflower, not organic broccoli. The top graph, this is the number of insects per 10 plant sample, and these were all collected from soil samples. You can see the cabbage maggot densities, early, mid, and at harvest. The rove beetle, which increased through the year. The number of spiders, and then the total predators if you add those up. That present parasitism from field-collected cabbage maggot pupae, the braconids, and the rove beetles. With respect to cabbage root fly biocontrol, we really have many, many, many more questions than we do answers. We're just getting started. So first and foremost, we need to characterize the parasitoid community. So what is it? When is it active? How does it behave? How do these BC contributions relate to when the farmers really need it? So in other words, early in the growing season, there's not a lot of biocontrol present. There's some predation, but there doesn't seem to be very much parasitism happening, and that's when the plants are most susceptible to cabbage maggot damage. And so there's a little bit of a disconnect there. And is there any way for conservation biocontrol to play a role in this sort of management, particularly if the chemical options for organic growers are very limited? I'd like to thank the funding agencies that help support this work. And do you have any questions?
Well, thank you. We're about to begin our question and answer session. So for anyone who missed the very beginning of the presentation, you can use the question box on your GoToWebinar control panel to type in questions and hit return. If you don't see the question box, you can click the small plus sign next to the word question to open it up. On Macs, I think it's a triangle instead of a plus sign. So I also wanted to mention before we get to our questions that we really value your feedback. So we would very much appreciate it if you could fill out the follow-up survey, which you'll be receiving in an email later today. Also, this webinar was recorded, and you'll be able to find it on our website and on the eOrganic YouTube channel if you'd like to see part of it again. If after the webinar you have additional questions that are more general about organic farming, you're also very welcome to use the eXtension Ask an Expert service. So I put the link for that on the screen too. So feel free to type in questions. We don't have a huge audience, so chances are your question may very well be answered if you type in a question. So first we got a comment here that it's not a question, but yes to charismatic maggots. So someone very much liked that. I liked it too.
I mean, it sounds like a bizarre statement unless you've actually been out there, and they're very friendly. They're very nice.
Okay. Well, let's see. With this is a general question about biological pest control. So with biological pest control, there are no chemical residues left in the soil or crop afterwards. Is that correct?
Correct.
Okay. Let's see. Cabbage worms are very present in my kale. Other than hand picking, what is possible?
Imported cabbage worms?
Looks that way, yeah.
Yeah. So there is a fairly robust parasitoid community all over that's pretty good at knocking these pests down. It really becomes a bit of a balancing act. And I haven't studied them exclusively, but I've been around long enough to know. So BT is very effective, right? The organically compliant larvicides are very, very effective at knocking down imported cabbage worm and all of the cole crop caterpillar pests. And so that's certainly an option. You should weigh that, however, with its ability to knock down syrphid larvae too, right? I mean, so if you aren't experiencing a lot of cabbage aphid pressure, then it seems very reasonable to apply an organically compliant larvicide. If you are, however, relying on syrphid larvae to clean up your aphids, then be really careful with applying a larvicide. With kale, yeah, that's a little tricky because you need the whole, it's not like broccoli where you only need the floret. In plants where you only are interested in harvesting a portion like a floret, it's almost just the frass you have to worry about, sort of the fecal matter that could contaminate the florette, because they rarely actually get in the florette. They're pretty happy with the leaves. But in something like kale, that's a little bit of a trickier concern. So you can either rely on parasitoids, which will do pretty well, although heading into the winter, or in December, I guess we're in December now. I don't know how much BC is going to be available in December, so you can collect the caterpillar, rear it out, and see if you get any parasitoids that emerge. It's pretty easy to do. Organically compliant larvicides are very effective too.
Okay, here's a question about whether any of these biocontrols are applicable to the northeastern U.S. as well.
I believe they are. So a lot of the syrphid species, at least Alligrapta obliqua, Eupiodes americanus, they definitely are there. And any syrphid species that I describe that's specific to the West Coast, there are going to be others. They're going to be present on the East Coast. Diadegma ancillare is on, which is the Diamondback Moth parasitoid. They're on the East Coast. And Cabbage, who knows? I mean, there's certainly some parasitoids there. But we don't know enough yet to. I don't even know the Braconid I have yet. There are certainly biocontrol agents, either generalists or specialists. There, I'm not exactly sure what they would be though.
Okay, here's a question about whether syrphid flies are available to purchase and introduce into a screen cage or greenhouse, and do you have any experience with the effectiveness of this?
I think just about anything is available for commercial purchase. It's just a matter of whether or not they're affordable. I am not sure they would be really affordable for a large sort of commercial scale operation. I think you'd almost be better off. That was indoor, right?
Yeah, it seems like he's talking about a screen cage or greenhouse.
Yeah. You can look into it. I mean, there are certainly some BC companies that sell predators. I haven't heard of anyone purchasing syrphid flies because they're probably too expensive.
Okay. Here's a question. How on earth do braconids find their root maggot hosts below ground?
Yeah, I know. That's a very interesting question. So we just started working on this, just started, and so I can't say for sure. I mean, I can't imagine it's the adult that goes down and risks damaging the wings to dig in the soil. So my hunch is that they lay an egg at the soil surface, and then that little larvae becomes an ectoparasitoid that goes down and finds the larvae in the soil. That's my guess. Because I mean, I can't see an adult parasitoid doing that.
Okay. Let's see. Do you have any recommendations on dealing with Bagrada bugs?
Yeah. So I've worked a little bit with them. I think there's some refinements in the organically compliant chemical control protocols that are being developed, and I think those are out. The initial word on the street, if you will, is that some of the trap crops are working pretty well. So Sweet Alyssum works as a preferred host. Those are some of the options. If this person's emailing from the Central Coast, there's a workshop in not this Friday, next Friday, about Brigada bug, I think in Monterey, which might be helpful. Yeah, so there's no BC agents that are available yet. I would say either look into some of the descriptions for the organically compliant insecticide protocols, or look into trap cropping, preferably on field edges. That at least anecdotally seems to be initially successful with a preferred host like Sweet Alyssum.
I guess somebody is emailing from LA and wants to know whether that seminar might be webcast.
The Brigada, I'm not sure.
Okay.
I kind of doubt it, but I'm not sure.
Okay. Let's see. Next question. Do you find a difference in parasitism rates in fields of cole crops which are planted sequentially? In other words, always cole crops in various stages in the same location versus fields of cole crops that are separate from one another?
I don't have any data on this, but I would imagine there would be. I know that I've seen cabbage aphid, for example. So there's one field of broccoli that just gets hammered with cabbage aphid. The syrphid plants, I'm sorry, the syrphid flies didn't quite get there in time to really manage the fast-growing aphid population. So they harvest that broccoli field, and then they plant another broccoli field right next to it, adjacent to that infested field. And the aphids immediately move next door, but so do the syrphids immediately move next door. And so they really do a wonderful job of managing that pest population in the second field. Given that this is a single community of pests, predators, and parasitoids, even if they're not specialists, even if they tend to be generalists, at least the good bugs in this case, they sort of act as a single community. If you kind of keep them all in one place, they're going to more reliably be there. That means the pest pressure is going to build up too. But yeah, my guess is yes. I guess that's a long-winded way of saying yes. I think they would be. Not without some challenges to go along with that. Obviously, crop rotation has a whole suite of benefits that are very well established. But just in the sense of maintaining continuity in your beneficial insect community, yeah, I would think so.
Okay. Has any other work been done with intentionally inoculating a crop with cabbage aphids early in the season?
No. I think no.
Okay.
That's where the work for alternate aphid hosts would come in. So you can imagine some kind of beneficial insectary that also provides a host feeding opportunities for some aphid that wasn't a pestin. So some aphid that's not cabbage aphid. The syrphids would come in because they're all generalists. They would start feeding on this other aphid early in the season, or before the season even started, build up their population, so that when cabbage aphid in the field actually became a problem, you'd already have sort of access to this really big, robust population of predators. They could just sort of go next door and go to work. So that's kind of the idea. I mean, that and cabbage aphids don't seem to have a hard time inoculating broccoli fields early in the season by themselves. They're so good, especially in the summer. They're so good at finding their way immediately to any cole crop that gets planted. Yeah, I think conceptually I love that idea. I think practically most growers would look at you crazy. But yeah.
Okay. Have you had any interactions with large yellow underwing moth caterpillar / cutworm? Is there any research out there about how to control them?
No, large yellow underwing.
Okay. Well,
caterpillar. I'm trying to think. I don't know exactly what you're referring to.
Okay. If you have any more information about the exact name of the species, that might help there. Okay. Are there any considerations for utilizing biological beneficial insects on GMO crops?
Boy, not an area of expertise.
Okay.
I mean, but I think some of the work. Boy, I'm gonna just. I think the predator parasitoid community can provide some benefits regardless of the cropping type. I mean, there may or may not be some implications for
Okay.
some kind of exposure, but yeah.
Okay. Sorry. Any suggestions for a good crop to help build up those aphids predator populations outside the field?
Yeah, so that's a great question. I haven't gotten that far yet. There'd be some considerations that you'd want to keep in mind, right? You'd want to make sure that there's absolutely no chance that the aphids that you'd be introducing would feed on your cash crop, your cole crop in this case. You'd want to make sure that the syrphids that are found in your crop are going to eat those other aphids. You'd want to make sure that the competition that's being presented by this other plant wouldn't be too intense. In other words, it's not going to set seed and start volunteering in your fields and become a weed pressure. There's no disease issues. In other words, it's not going to be a vector for a particular plant pathogen that could get crossed over to the cole crop. That it works in the same soils, in the same temperature range. So there are lots of there's a big checklist of things that you need to think about. And I would love to do that work. I just haven't gotten that far yet. But you can imagine once you start thinking about it, okay, you got to figure this out, and then this, and then this, and then this, and find the right candidate for your particular spot.
Okay, someone very helpfully posted the link to the Bagrata workshop, so I'm going to send that to everyone. And it says in that it is actually going to be broadcast. So
fantastic.
Yeah,
great.
Thank you very much for that. I just sent that in the question or chat box there. So hopefully it's a really long link, but I would suggest maybe Googling instead of trying to frantically type down that endless link. You might want to Google CCOF Bagrata Bug Workshop because it seems like CCOF, California Certified Organic Farmers, seems to be organizing that. Is that correct? Looks that way. Yeah, or whether it's not, it was in their weekly news from November 23rd, 2015. So that should get you started with the search engine there. Otherwise, you're going to have to go after this link, which is kind of huge. So yeah, maybe Google CCOF Bagrata Bug Workshop and see how far you get there. Hopefully you'll get there and be able to connect. Thank you. This person is wondering about ladybugs and whether they can be used to control aphid infestation.
Yeah, ladybugs are, famously known as really successful aphid predators. The extent to which they come and consume cabbage aphids, I don't see it that often. I'm not sure. Cabbage aphids are pretty high in glucosinolates and all these sort of defensive compounds that aren't very tasty for a lot of insects. So that deters some generalist predators from gobbling them up. I've never seen anyone buy ladybugs and release them on a commercial scale for cabbage aphid management. Maybe indoors you could, or on a small scale possibly, but I'm not sure that'd be the best fit in a lot of cases, but maybe. Yeah.
Okay. This person says, my main predator is paper wasps. The aphid outbreak is after the wasps hibernate. Okay.
Yeah, I have heard of this. So paper wasps and other wasps were really great predators, and they can, suppress a lot of pest populations. I would say just in general, during the winter, a lot of biological control agents are not very active at all, and so this is when you start to worry about big aphid outbreaks because you just don't get. You have sort of the seasonal suppression of good bugs that are available to help you out, and that's when you kind of see the sheer reproductive power of cabbage aphids that is sort of unadulterated, and it gets. This is when I would start to be a lot more proactive with organically compliant aphid sprays, because I don't. In the winter when it's super cold, you can't anticipate a lot of help from the good bugs.
Okay, here's a question about where and how do syrphid flies overwinter, and also the I don't know how to pronounce it, ichneumonidia wasp. That's a hard word.
Yeah, ichneumonidae.
Sorry. Okay.
Yeah. Let's see. So with the ichneumonids, I believe it's the soil surface, but I don't recall for sure. With syrphids, I've got to admit, I also don't recall whether or not what the diapause or overwintering strategies are. Well, that's a hole in my knowledge. I'm not sure.
Okay.
That's a good question. But I would, so a lot of the kind of protected habitats, hedgerows, shelter banks, those sorts of things seem like a good idea, just because regardless of what it is, they've got to find a stable and undisturbed overwintering spot, and that would be a good resource regardless.
Okay. Here's a question about whether or not you're having trouble with whiteflies.
We don't tend to, no, not in organic coal crops. No, I think that just working in organic coal crops and organic strawberry, the whitefly stuff tends to be a secondary pest outbreak resulting in lots of sprays in conventional ag, but I've never seen it as a primary or sort of key pest in outdoor organics. Now, indoor organics and indoor with other crops or organic, there are other crops that are certainly susceptible, but I have not seen it in coal crops, organic coal crops, no.
Okay, I think that was the last question that we've gotten. And I just wanted to remind everyone that if you're interested in coal crop pests, we're having another webinar next week on December 9th. It's from a different region. It's from Auburn University and the University of Florida, so Alabama and Florida. But they're going to be talking about yellow margin leaf beetles, which is a pest in coal crops down there. So if you're interested, feel free to sign up. You can find the upcoming and also the archived webinars at the link there on your screen. So that looks like that's it for the questions. So I'd like to thank everyone for your questions as well as the information on the Grotto Bug Workshop, and encourage you to check out our many other upcoming and archived webinars on organic farming and research topics at the link on your screen. And again, we'd also appreciate it if you could fill out the survey that you'll be receiving in an email later today. Thank you so much, Diego, for sharing your work with us today, and thanks to everyone for joining us.
Thank you.